Katsikini from the Aristotle University of Thessaloniki, and I will chair the session, which has the title Biomedical Imaging and Cultural Heritage. The first talk is going to be given by Paul Tafferot, and the title of the talk is From Cultural Heritage to Biomedical Imaging, Development of Synchrotron Phase Contrast, Hierarchical Imaging at the ESRF. So Paul, please start.
Thanks. Good morning to everybody. So I hope it works. So today I will present you the application of phase contrast microtomography at the ESRF, and especially in the case of cultural heritage, paleontology, and how it evolved into biomedical imaging recently. So for that, I don't know if all of you is familiar with the technique of tomography, but typically tomography, it means making slices in a sample and make picture of these slices. So basically what you have on the screen is a tomography. It's a physical tomography. But when you are using X-rays, and especially when you are using a synchrotron, you can do X-ray tomography, meaning 3D imaging of the internal structure of an object without destroying it. So it's a main interest in X-ray tomography. It is the fact that it is non-destructive. So most of the tomography worldwide is done with laboratory machines, but when the laboratory machines are not enough, you can go to the synchrotrons that are really the golden standard to make the high quality tomography in complex objects. And there are several reasons for that. So I will show you a few examples. Just to give you a bit of background, originally, I am paleontologist, so working mostly on fossils, but I will show you how all the techniques developed in tomography to image plenty of things and also to image fossils have been put together during the COVID crisis in order to push the biomedical imaging to a new level. So I will not go in very much details of physics and so on, because I'm not a physician at all. I will just show you the very key point that explains the success of tomography at the ESRF and in synchrotron in general. And one of the most important one, I would even say the most important one, is the use of phase contrast. So what you see for the moment, it's a piece of opaque amber. So amber is fossil tree resin. This one is about 90 million years. And if you are using X-ray absorption contrast, so basically the same kind of absorption of contrast that what you can have with laboratory machine or with medical X-rays, with absorption contrast, you see nearly nothing in such kind of specimen. But when you are using a third generation synchrotron, and even more a fourth generation synchrotron, the beam, when it goes through the sample, it's not only absorbed by the sample. The wavefront of the beam is distorted by the sample, and this distortion creates the phase contrast effect. You have different techniques to see the phase contrast, but the easiest one that is very easy to apply with such kind of synchrotron sources, it is the propagation phase contrast. You just increase the distance between the sample and the detector, and just by doing that, by changing the distance, you have the propagation phase contrast effect. It is what you see right now. By increasing this distance, we increase a lot the sensitivity to the small density differences. And in this case, you can clearly see the two fossil insects. So when you go in 3 dimensions, you can do that with phase contrast microtomography. And here are 2 examples of fossil insects from Cretaceous. So the imaging of amber is something quite easy because it's low density. Let's go to something a bit more dense. What you have there, it's a small fossil egg from Thailand, again from Cretaceous, from Thailand. And in this fossil egg, there were some information that there could be fossilized embryos, so fossil bones. We made conventional absorption microtomography. So it is what you have on this slide. And on the slide, you can see some white structures that are bones, fossil bones, but that are very difficult to see. And you can see only very few of them. And again, we just increase the propagation distance by 1 meter. And thanks to the propagation phase contrast, you have an increase of sensitivity by a factor 1000. And you can see that all these black structures everywhere are fossil bones. So basically, you have complete fossil embryo in this egg. So if you see that in 3 dimensions, here you have the laboratory CT. Here you have a synchrotron, but in absorption mode. And the last one is synchrotron with propagation phase contrast. You can clearly see why it is making a so big difference for X-ray imaging on difficult samples. So these 2 examples were on quite small samples. Let's go to something much larger. Here you have an hominid skull from South Africa that is about 15 centimeters in diameter. And it was imaged originally with a medical CT. So what you have there is the result of the medical CT. And what we managed to do in 2010, so it's already quite a long time ago, on ID17 at this time, using specific scanning protocol for large objects. And you can see that the increase in quality is quite visible. The resolution is much better. But for us, this kind of resolution, we are at 45 microns right now, is a low resolution. It's really the lowest resolution we are doing. So again, when you go in 3 dimensions, thanks to the synchrotron data, it was possible to extract the endocast to have information about the brain evolution. So, and now what is really the second key point of all that, it is the fact that you can go with multi-resolution. So you start with a large object like this skull, and then you zoom in and you are able to reach very high resolution in small parts of the objects. So we have several examples. The first one, it's a small fossil ape from Kenya. The fossil is about this size, about the size of a lemon. And you can see that we made the full scan of the skull at 30 microns. Then we see the teeth at 13 microns and single tooth at 6.5. Then we go to 2.2 microns. At this level of resolution, you can see the incremental lines indenting. And then we go to 0.7 microns. So basically the resolution you would have with a microscope. And in this case, you can see the daily lines recorded in the enamel. So like that, it was possible to calculate the age at death of this young ape. Even if it is 13 million years old, we were able to calculate the age at death with a precision of something like 40 days. So you see there one of the applications of the multi-resolutions. There are other ones on even larger specimen. So this one, it's a complete dinosaur from Mongolia that is 74 million years. And the specimen is about this size. So it's about 50 centimeters. We scanned completely at 45 microns. And then you can see we went to see details like the snout at 6.5 microns or the teeth at 2.2 microns. And for some parts of the bones, we even went to 0.7 microns to see bones microstructure. Again, all that without destroying anything on the sample. The key point there is always the non-destructivity. Third example, much more recent, it is on an Egyptian mummy from Ptolemaic period. So it's a crocodile mummy that is 1.1 meter in length. And again, we made the full scan at 90 microns. Then inside the crocodile stomach, we discovered a complete mouse that we scanned at 13 microns. And we increase again the resolution to see the details of the teeth of the mouse at 3 microns to be able to know what genus of mouse it is. So now we know that it's a genus Mus. So by making the full study of this specimen, we demonstrated that it was a wild crocodile that was hunted to make the mummy. And an example, when you go in animation mode, it's another crocodile mummy. I'm sorry, it seems not working so well. We are at 40 microns for the full imaging. Then we remove the balm, we remove the piece of fabric. And inside you can see that all the skeleton have been removed and you have plants everywhere. And now we are at 4 microns in pixel size. At 4 microns, we are able to see all the details that would make possible a precise determination in botanic. And as I said, there is no skeleton in this one. It has been basically completely emptied. And if you go to the fingers, you can see that the bones have been cut and we can study the cut marks non-destructively without having to open the mummy. And again, trying to understand what happened, why there is no skeleton, because in all the other ones, there is a skeleton. We went in the skull, and in the skull, we discovered a lot of remains of insects. And this one, it's a dermest that has been scanned at 40 microns, 20 microns, and 6 microns on the right. And this one is an insect that is eating fresh cadavers. So what we understood is this crocodile was not raised or was not hunted to make the mummy. It was a cadaver discovered somewhere that was already in advanced state of putrefaction. And the ancient Egyptians just took it, remove everything that can rot in the cadaver, remove the flesh, remove all the skeleton, and you just keep the things you cannot remove without harming the without damaging the skin. So you have to leave the skull and the fingers. Okay, so take all that into account: the phase contrast, the multi-resolution, the imaging of large objects. You go in beginning of 2020 with the rising of the COVID-19 crisis. And at this moment, ESRF was approached by a group of people, so German medics, Maximilian Ackermann and Danny Janik, who contacted Peter Lee at the UCL. And their team contacted the ESRF to know if somebody would be able to scan complete human organs and zoom in order to understand the effect of the COVID. At this time, it was very difficult to understand the effect of the COVID on the complete organ. They had a lot of information from medical CT on one side. They had also a lot of information from histology, but no way to relate the two things. It's one of the common problem in biomedical imaging. You have the two extremes, but it's very difficult to link the scales together. So if they would have come before the EBS source, we would probably have said that no, it's not really feasible. The contrast is not good enough. But just before the lockdown, we had few weeks to test the capabilities of the new source, the EBS source. And we made this test on BM5, that is a beamline, one of the historical beamline of the ESRF, where we are doing tomography. It is the one we use to make the crocodile mummies, for instance. And because we had these few weeks to test before the first lockdown, we said, okay, it should be feasible. Let's give a try. And then we prepared everything to go to imaging of complete organs by combining all the techniques basically that were applied to fossils. So if you take all these techniques, you can have simple phase contrast where it's very difficult to see anything because we are in soft tissue organs. So there is basically no real contrast. Then if you make a specific attenuation protocol that is derived from imaging of large fossils, you can have already much better results. And finally, you increase the dynamic by using the accumulation protocol, and you can have high quality scans in a complete organ. So right now we are at 25 microns. And then we pushed the system to go in hierarchical mode, to go to higher resolution. So it is the last step, the hierarchical imaging, and you have this transect at 2.5 microns. At 2.5 microns, you can see that you have very high resolution in the organ, again, without cutting anything in the organ. And on this specific one, we extracted after the scan the piece that was scanned, and we compared with classical histology. The top line, it is the synchrotron imaging in the complete organ. The bottom one, it is the histology made on a small part of it. And we just use false color to make possible the comparison. And basically, what you can see is a perfect match between the synchrotron scanning and the histology. So, as I said, we are in a hierarchical aspect, and the main question originally was to understand the effect of COVID on human organs. So this one is the very first COVID lung we scanned for this project. It's an upper lobe. Oh, sorry, it's not working. Okay, so we started at 25 microns. Now we are at 6.5 microns, and you can see a part where all the alveolars are in good contact. It's a part where the air is still able to work well, and another part where all the alveolars are separated. It is a part where alveolars have been filled by mucus or by fibrosis, and that is not functional anymore. And then we zoom in again, and we see the effect of COVID. I'm sorry, it's really not working. And we see the effect of COVID in the vascular tree. So with general thrombosis, so coagulation in the blood vessels, everything that is yellow is basically completely blocked. And we also see hemorrhage in alveolars and so on. So basically what we discovered with all these scans, it is the fact that the COVID is affecting the lungs in very different ways. First, it is degrading a lot the airways. So a lot of the airways are just completely blocked by mucus or by fibrosis. But on top of that, the second effect of COVID on lungs that is also affecting other organs, it's not on airways, it is on the vascular system. So if you look in the same sample, you go to 6 microns and you look at the vascular tree. So in blue, you have one of the air conduction system. In red, you have the arteries. In yellow, you have all the capillaries and all the small vessels. And in fact, on this picture, you have way too much vessels. You have to know that in lungs, you have two different vascular systems. You have the vascular system that brings the blood to the alveolars to exchange the gas, where you exchange the CO2 for the oxygen. And you have the second one that is the blood supply of the lung itself to keep the organ alive. And normally, these two systems are completely separated. And what we discovered with these scans is the fact that in case of COVID-19, you have neoangiogenesis. So it means new blood vessels that are growing in a kind of hieratic way. And what happens is you have anastomosis created between the two circulation systems. So it means that two blood systems that should be independent are not independent anymore. And you have anastomosis like this one, where you can see that the blood, instead of going to the oxygenation with the alveolars, will directly go in the second circulatory system. And so even if the volume of air in the lung was still okay, and even with pure oxygen, the oxygenation of the blood was very bad because the blood was just making a shortcut and not going to the alveolars. So it is one of the explanations of the bad effect of COVID. So we started with the COVID, but very rapidly we understood that this technique would have a huge implication for imaging of in biomedical in general. And so we started to work on other organs. So what you have there is a brain image at 25 microns. Now we are at 6.5 microns in the cerebellum, and then we push to 2.5 microns, again with this hierarchical logic. And at 2.5 microns, you start to see some cells and you see very well all the microvascular things. I'm really sorry for the movies. They were working perfectly this morning, but not today. So with this hierarchical imaging, you have the possibility to reach the level normally you would have with histology, but with a complete context of the organ. So you can relate all the scales and go from the full organ to the histological level. And you will see later today that if you work on small parts of the brain, then you can go much, much further by using other techniques like nanotomography. So all this project, so far we imaged something like 30 organs, and we rapidly realized that it was important to have this project growing because data were interesting for many people. So with funding from the Chan Zuckerberg Initiative and with rapidly growing collaborative network, we started the Human Organ Atlas that is hosted at the ESRF, and we put in the public domain all the tomographic data from these organs. So for the moment, we have 6 organs in this atlas. We will that are control organs plus 1 from the COVID. So the lung I was showing with all the COVID aspects, all these data are public. And we will put normally 6 new ones in the coming weeks. We are just starting a second batch of data release. So it's open access. You can just go on the site and download what you want to study the data. Just be careful, the data are quite large. For some of the organs, like the lung you have there, the total data is 6 terabytes. So all the things I showed you were done on BM5, started during the COVID crisis and so on. But in parallel of that, we are building a new beamline that is called BM18, that is also known as EBSL3 because it's one of the EBS beamline. And this beamline is basically the same principle, but much larger with much higher coherence. It's taking full advantage of the new source of the ESRF. So it is dedicated for hierarchical tomography using mostly propagation phase contrast imaging. And the big difference is we can go to high energy. So for the moment, we are at 220. We expect to be able to reach 300 kiloelectron-volts by next year. It is the longest beamline at the ESRF for the moment. We can go to 40 meters of propagation. To give you an idea, what you had on BM5 was 3.5 meters. And this propagation distance gives you basically the maximum contrast you can have. And in terms of sample size, on BM5, we can do samples up to 30 kilograms. On BM18, it will be 300 kilograms. So it's really a very different scale. And as I said, this beamline has been done to take full benefit of the coherence of the new source. So it is the very large beam with very high coherence at high energy. So it's something that is really unique. The 5 main topics of BM18 are the biomedical imaging, as I just explained, but also natural and cultural heritage with paleontology, archaeology. But it's also a beamline that is the largest part is material sciences. So you can be batteries, metallic alloys, can be additive manufacturing, plenty of things. We have a large part in industrial applications. It's a beamline that is really interesting for many industrial applications. So we are already started in this industrial part. And we have also a project on geology. But as explained by the directors before, these 5 topics are the main one. Does not mean that you cannot go in other topics. Basically, everything that can be interesting to scan with microtomography has a good reason to go on BM18 if you cannot work with conventional CT. So the picture you have there, it is the beam of BM18 in the experimental hutch a few months ago. And normally the X-rays are not visible, but as we are using wide beam with quite a lot of power, you have ionization of air, and that's why we can see the beam. So it was for the radiation test, we are never using so much power for imaging of real objects, evidently. So why is it making a difference to use BM18? So here you have again a human brain. On the left, you have an MRI done with at the hospital. In the middle, you have the BM5. So basically the same kind of thing that what I showed you. It's another brain, but it's the same data quality. And on the right, it is BM18 with 31 meters of propagation. So when you compare between BM5 and BM18, it is clear that the sensitivity is much higher on BM18. So it is really the key point of this beamline. We push so much in terms of coherence and in terms of propagation distance that we can reach what is really the limit in terms of sensitivity with propagation phase contrast. So you can see the level of detection is completely different. So that's why we are expecting really a completely new level in biomedical imaging, but not only biomedical imaging. So this project on organs is growing very rapidly. And as I said, we are working on other organs. So for instance, here you have again a brain. And in this brain, we discovered the beta amyloid plaques. So we were able to see one of the aspects of the Alzheimer's disease in the complete brain. Here you have a prostate with calcifications and with some defects in the organ. We are also working on the heart. So what you have there is a kidney. So for the moment, BM18 is still using a small tomograph. So we are using a machine like this one. It is a small tomograph like the same one than on BM5. But hopefully by autumn or let's say beginning of winter, we should have the final sample stage of BM18, that is this gigantic system. So basically, we want to go from 30 organs per year to something like 200 or 300 organs per year. And the final step will be to go to imaging of complete human bodies with the quality from the synchrotron phase contrast. The idea being that we will use the data from the synchrotron to train algorithm to make medical machine more efficient by comparing the data from the same body with medical imaging CT and with the synchrotron imaging. So with that, I thank you for your attention. There would be way too many people to thanks for all the things I showed, so it's nearly impossible to do it. So thanks to many people. And if you have questions, just ask.
Thank you very much, Dr. Afero. Are there any questions from the audience? Thank you very much, Paul, for a brilliant lecture, which is quite fascinating, to be honest.
I don't know if you can hear me.
Yes, yes.
Yes. I will transfer a question from the audience that maybe Maria would like to come up and explain better the specific project.
Thank you very much for your talk and the fascinating photos. So my question, I'll try to articulate it as best as I can because I'm really not familiar with any of these wonderful things that you're doing. So I'm a biologist and I'm working on stress biology and cryptobiosis. And I have as model organisms, they call tardigrades. Their common name are water bears. I'm not sure if you have ever heard about them. So
Yeah, I know that. Yeah.
okay, so they're minute organisms. I'm just saying a few things for the audience. Around 100 microns large. And the cool thing about them is that they can endure several kinds of stressors and extreme stresses. So they're not extremophiles, but they are the best extremotolerant organisms that we know. So my question is, I have seen through transcriptome analysis that IDPs, intrinsically disordered proteins, they're key players in making these organisms tolerant as well as they are. Would it be possible to use live organisms in order to analyze it under the beamline and have some data on the IDPs?
I see two main problems. The first one is the size of the organism because they are very small, so it would be adapted for nanotomography, not really for microtomography.
Mm-hmm.
And even if they are very resistant, I doubt that the high X-ray dose that is required for this technique, so either microtomography or nanotomography, I doubt that it can be compatible with in vivo. Or maybe it would work for very, very short time, let's say. So everything, it would depend on if you already have some knowledge about what is the maximum dose these animals can handle.
So what we know is that they have survived cosmic radiations because they have taken the stressed organisms. So they go in kind of like a cyst when they are upon stress. And this is how they endure the stress. And and then when you put them back in normal conditions, they have retracted their legs, so the legs and the head pops up again. And they continue as usual, like nothing happened. So they have put them in open space conditions back in 2007 or 11. Forgive me about that, I'm not remembering well. And also they have endured the whole process of the electron microscopy.
Ah, okay, so then maybe, yes, from what you described, it should be quite close to what they are doing on ID16 when they do cryo nanotomography in vacuum. So maybe, clearly it's not for BM18. BM18 is done for quite larger things, but I think for ID16, it has to be discussed with Alexandra. I see that she's online already. So it's more for ID16.
Okay. So it's not a no to begin with, right?
I would say it's a maybe. I don't know. Some calculation are needed. Let's say.
Okay. Thank you so much.
Okay. Are there any questions?
Yes. Yeah, there is one. Excuse me. Yes, there is one from chat. It's from Alexandra Frankovich. Excuse me. I apologize if I pronounced that wrongly. So the question is regarding the multi-scale logic. Is it all done within one acquisition, or do you acquire one complete scan at low resolution, then another of a smaller area at higher area, etc.?
So in fact, we can do both. For the moment, it is more the logic where you do a first scan at low resolution and then you do other scans at higher resolution. So this one is really the core aspect of BM18. But by next year, we will have also a detector with a very large number of pixels, being able to make single slice of 32,000 pixels. So basically, in this case, if you use the full resolution of the detector, you make a multi-resolution reconstruction, meaning that you scan the complete object with the full data, but the volume is so large that you cannot really work with the full resolution. So you reconstruct it at lower resolution, and then from that, you select what part you want to see at higher resolution. So in this case, everything has been scanned only once. But I would say the vast majority of the experiments will be done in a sequential way, where you do the first complete scan at low resolution, and then you select where you want to go with the higher different resolutions. So BM18 on this respect will be highly automatized. We are just finishing the installation of our detector stage, where we will have 9 different detectors that can be automatically exchanged to change the resolution and to go from the largest one to the smallest one. At the end, we should cover something like from 0.5 microns to 200 microns in terms of resolution range.
Okay, the resolution was impressive, and the details you can obtain these pictures. I want to ask a few questions. How do you stabilize the sample? How do you fix the sample? Because I understand that the sample should be very stable in order to record such pictures, such scans.
Yeah, in fact, for imaging of organs, sample mounting is 90% of the job. It's really the most critical part. And so we took several months to develop the protocol of mounting. It is, so the organs are prepared, they are fixed in formalin first, and they are prepared with ethanol up to 70% of concentration. And after that, they are mounted in plastic jar using agar gel that is also at 70% of ethanol. So you cannot make directly agar gel with ethanol. You have to make it with water, and then you equilibrate with ethanol. And we make small parts of this agar gel that we just pack all around the organ. And once it's fully done and we remove all the dissolved gas and so on, we have a way of mounting that is extremely precise and that can be stable. So far, we rescan the same organs for 1.5 years now, and we are still able to relate the different scales after 1.5 years. So, but yes, the sample preparation is really the most critical part of all the process. I mean, the imaging itself is evidently quite difficult. But if the mounting is not perfect, there is no way to make the kind of picture I showed you.
Okay, the next question is, can you simultaneously record information from X-ray fluorescence? I'm
sorry, can you repeat?
Can you simultaneously with imaging, with phase contrast imaging, obtain information with X-ray fluorescence, with XRF? On the content of the metallic elements which are contained in these tissues?
Not on BM18. BM18 was really optimized for high energies where the X-ray fluorescence is quite difficult to use. But other beamlines can do that. And again, ID16 A and B are able to make the two techniques in parallel. I think also ID15 is developing something like that. So in principle, yes. In practice, I don't know if it is often used or if it is done more in a sequential way, because you have different ways to make the X-ray fluorescence. In many cases, you have to go with a focused beam. And in this case, it's not compatible with the phase contrast imaging in the same time. So the examples I know, but again, it has to be discussed with the other people from ESRF. Typically, it goes in sequential way where you do the imaging on one side and the fluorescence on the other one. But technically speaking, it's possible to do both.
Okay, thank you very much. So the title of the next talk is Hard X-ray Bioimaging at the Nanoscale, which is going to be given by Dr. Pakourianou, if I pronounce it well, again from the ESRF.
Okay, so I'll try to give you an idea of the capabilities in terms of nano imaging for life sciences. And I'll show mainly applications from the beamline ID16A. So this comes a bit like quite naturally in continuation of micro-CT, so moving to the finer scales. And here I try to just quickly highlight the main points why using X-ray microscopy at the nanoscale is really a precious tool for exploring living matter. So this is starting with the fact that we can reach a resolution in the range of a few tens of nanometers. And as Paul showed you, by using phase contrast, we can actually have extremely good contrast in unstained tissues and cells. And the fact that X-rays at high energy, they can penetrate samples means that we can image them without sectioning, that is the case with most of the other microscopy technologies. And we can combine this with cryogenic imaging, so we can keep the samples close to the native state by vitrification. And with the high brilliance of the fourth generation sources, we can go quite fast, and then we can image multiple samples, so then you can see how organisms evolved with diseases or when they developed simply. And you can also combine it with other technologies, both before and after doing X-ray imaging. So this is just a picture of the end station, so the microscope in itself. So what I would highlight here is that we do use a nanofocus beam, and also that the system is fitted with a cryogenic system, so we can image the samples at minus 160 Celsius. And there are two technologies which are present together within the same end station, which means that once you put a sample in there, you can use both of the technologies. And on one hand, we use phase contrast tomography, in particular holography, and this gives you the nanostructure in 3D. And on the other hand, with X-ray fluorescence, we can reconstruct quantitative maps of trace elements with very high sensitivity, sub-ppm level. And this is like an example where using together these technologies was very useful to address a specific scientific question. So researchers were wondering how cells react to silver nanowires. So these are present in many electronic devices like a flexible touch screen. So we are getting more and more exposed to these nanowires, and we don't know what happens. Do they go inside our organism? How are the cells reacting? So we took cells in culture, we sprinkled these nanowires, which were, some of them were 30 nanometers diameter, some were 90 nanometers. And then we left them for a while to see how the cells interact. And then we froze the cells, and then we imaged them with phase contrast. And one of the questions was like, do they penetrate inside? And we see that indeed they do penetrate in the cells, but we also could see that the cells actually have sufficient mechanical force, at least when the diameter is quite fine, so 30 nanometers, to fold them. And they fold them and then they rub them in vesicles, so they try to isolate them. And on the other hand, by doing fluorescence maps, so you can see here maps from phosphorus, sulfur, silver, so you actually see the nanowires, zinc, and so on. We could see there is also like a self-defense mechanism based on chemistry that's activated, and notably that there is sulfur deposition on the wires and copper. And then for like staying a bit in the X-ray fluorescence part, one of the prominent applications is looking into drugs for different diseases, and there are quite a few in the area of cancers, of course. So for example, we looked, so typically what we try to do is to precisely identify which are the targets inside the cells of these drugs in order to understand their mechanism of action. So for an osmosenil tamoxifen, so this is a drug that one of the very few that are effective for triple negative breast cancer cells. We could see that it accumulates in the endomembrane system. Annalise already showed you a little bit earlier. And then in the case of malaria, we looked at chloroquine that now everyone knows about. And we could find that bromocrine caps the hemozoin tracer. So basically you can understand more how the actual drug acts and how it is effective. And then another example, again, in the case of chemotherapy drugs, iridium. So iridium, it's an anti-cancer agent that's quite powerful in different types of cancers. And by combining, and in this case it was soft X-ray tomography in ALBA with X-ray fluorescence here at ESRF, we could see that the drug goes exclusively in the mitochondria, and we could also precisely quantify it. So you see here, introduced with this mesh It's like it's wrapping the mitochondria, and then inside you see, following this color scale, the presence of the iridium. And then kind of moving to something that's a bit more like fundamental science, we combine these two technologies also to try to figure out how pollen grains, which are very small and very light, how do they protect the genetic material so well, even through mass extinction? So, for example, when the dinosaur disappears, pollen managed to preserve the genetic material of coniferous trees, for example. So, by the fact that we can uniquely combine cryogenic imaging at the nanoscale to see the structure, and also X-ray fluorescence, we could see on one hand that this outer part of the grain it's a nanofoam, and in fact this was the first nanofoam that we discovered naturally in made by nature. And we also saw that there is quite high concentrations of calcium in this exime part of the grain. And in fact, these pollen grains are used as a template for biomimetic materials, and also the pharmaceutical industry is trying to use this for drug delivery, so the information is useful for different communities. And now I'll focus more on the 3D imaging with phase contrast. So the kind of the advantage of this type of technology that we use, which is full field imaging using a focus beam. So we place the samples in the divergent beam. It's making it easy to image samples at different scales by simply changing the position of the sample between the focus and the detector. So we typically image at voxel sizes between 10 and around 100 nanometers. And just to give you an idea, so the raw data of a simple object, as you see on the lower right side, it looks like this, a bit psychedelic when you place the object at different distances between the focus and the detector. And then, so this kind of imaging, it's of course very well suited for kind of hard materials, so for bones, but also for nacre. And the fact that you can resolve things at very fine scales, such as organic interfaces among aragonite tablets in nacre, enabled us to discover new things related to morphogenesis, and notably to try to figure out how nature generates highly ordered structures. But then, of course, more on the kind of biomedical side, it's very interesting to look at soft tissues. And here you see two examples of soft tissue imaging without any staining. So there's lung and heart. And actually the lung tissue is also from a patient which had died of COVID. And we see at even like more detailed and finer scale how the tissue is clogged with all kinds of residues and so on. And on the right, you see the heart muscle, so you can resolve quite well the muscle fibers. Yeah, and then if you look in 3D at some pieces of the lung, for example, and you look at one from a healthy lung and one piece from a lung with COVID, you also can visualize how tissue is really clogged. And also we can look with fluorescence to see what are the elements present in there. So we see quite a lot of minerals, so calcium combined with zinc and phosphorus, but also iron. And now I'll talk a little bit more about neuroimaging. So in this field, the X-ray nanotomography can have a really large impact, notably because we are trying to reach both resolution and the scale where we can look comprehensively at the neuronal circuits, that is to see all the neurons and the connections between them in full brains, or to see it in volumes which are large enough to be somehow independent. So starting from very fundamental questions like how can a collection of interconnected cells give rise to intelligence or behavior, to how can we use or better understand how the brain works and translate this to either chips. So there's this new generations of neuromorphic chips, but also robots. Everyone heard about Boston Dynamics. How can we translate this to technology? And of course, what concerns every of us, it's also neurological diseases, which we can understand yet very little, and we don't have effective ways of coping with them. And here there's like a quite great challenge of scale, in the sense that if you want to image full brains with sufficient resolutions, until now you could only do it with electron microscopy by sectioning the tissue very like with a few tens of nanometer thick sections, and then imaging this, so it makes you millions or tens of millions of images that you then need to kind of stitch together in a big 3D image. And this is very tedious. So the first brain map took about 20 years to make, starting from the 80s. Recently, a brain map of a fruit fly has been achieved, but it took about 5 years of data collection, and it was very expensive, as you can see, like tens of millions of euros. And then the next big sort of challenge is to go to a mammalian brain, which is closer to ours, and there are many models for diseases and so on, so it would be very useful, but scale-wise, to something that's kind of easy to imagine, is like going from the width of a seat of an aircraft to going from Boston to Lisbon, so across the Atlantic Ocean. So in this context, what is nice with the X-ray microscopy is that we can fit, for example, this fruit fly brain in one field of view. So in a couple of hours, you have an image of a full brain instead of a few years. Of course, you need to zoom in and achieve higher resolution to actually trace the neurites and the connections. Then we actually did the exercise to image the same tissue region with both holography and electron microscopy, and we see that the contrast is quite similar. Nevertheless, we need a little bit more resolution with X-rays. And then here, just to make a connection with the postdoc that you see just earlier, so I took a small image from one of his publications, where you see like from a brain, a tissue region that you kind of zoom in to start to see first cells, and then if you look at this like little square, you start to see that actually there's more and more structure. More you zoom in, more you discover new structures. And even from here, if you further zoom in, you barely start to see the connections between the neurites. And actually, we could use this to already address some scientific questions. So in one case, we tried to gain insight into how walking is coordinated and controlled by the nervous system. So we looked at the motor neurons, which are connecting the spinal cord of the fruit fly to the leg, all the way to the muscle insertions. And what is nice also with X-ray imaging, that you see not only the neurons, but you also see the muscles. So you see all, and even other small organs that control coordination of locomotion. And then just a small parenthesis, in order to be able to extract useful knowledge from all this data, normally you have to go to segmentation, where you separate the structure, and this is not trivial. As you can see, the images or the structure itself is very complex and intricate, so we need to use automated image analysis technologies, which nowadays are based on deep learning mainly. And then I'll just quickly give you two examples of more applied applications closer to kind of clinics. So in one case, we look at tissue biopsies from patients which suffer from diabetes. So we looked at people that suffer from diabetes type 1 and 2. And they develop a neuropathy in their limbs, so they develop numbness and pain in their arms, for example. So we looked at fascicle of nerves from near the wrists, and what we saw is that the people with diabetes type 1 have the axons of the nerves are actually affected in the sense that some of them are much smaller. They have much less myelin, so myelin is insulating around the axon in order to favor transmission of electrical signals, and also that some of these axons are kind of wrapped, entangled around each other. So we get some insight into, okay, what is the relation between morphology and the symptoms. And then a lot of the work is done, of course, in model organisms. So for example, in a mouse model for multiple sclerosis, we could have a look at what happens when we try a treatment with mesenchymal stem cells. So in this case, we looked at tissue from the spinal cord. So people with multiple sclerosis, they usually end up in a wheelchair with them. And what happens is that the motor neurons and the blood vessels, so the motor neurons disappear basically, and the blood vessels also are very much affected. And we're quite happy to see that this treatment seems to be effective, because we saw that neuromotors were again present, and also the blood vessel capillaries were again well structured and regenerated basically. And then another kind of example where this kind of technology is useful, is to create ground truth data in order to design more accurate biophysical models for other technologies. So in this case, for functional MRI. So this is used as a diagnosis technology. So the patients can take it. There's no risk. However, this relies on assumptions that the axons of the white matter, where you have this kind of highways through the brains, the axons are always of constant diameter and going like in parallel bunches. And if you look at a finer scale, you actually see that this is not really true. So the diameter varies. There are trajectory deviations due to interactions with the cells in between. In some regions, the trajectory are really like quite entangled. So basically, we can create a data which can be used to define biophysical models which are more precise and use them in the hospital. And then like one final example on model organisms. So in this case, in a zebrafish model for autism spectrum disorder, we could actually image several full brains. So we have 6 brains of control, so healthy ones, and 6 brains of fish which exhibit this basically symptoms, especially in the sense of being hypersensitive to sound. And then we are still we just started to analyze the data basically to find out how the brain actors change in the context of autism spectrum disorder. And with this, I'll stop here, and I'm happy to answer questions if there are any.
Thank you very much, Dr. Pakourianou. Are there any questions from the audience?
Yes, actually, there is one from the chat from Manuela Filippidi. Ms. Filippidi, can you hear us?
Yeah, hi. Thank you very much for the informative talk. So I have 2 questions. So I'm at the University of Crete, by the way, and I work on biomaterials. So my first question is that on benchtop nano CTs, typically one would have to slice down the sample to maybe about like twice the field of view. So maximum would say like 50 microns, 70 microns. Is that something that in a synchrotron setup does not need to be made? So could the sample be in its original form, like larger?
Yes, thank you for the question. So yes, we typically work with samples which range from a few hundreds of microns to millimeter in size. So we can work with quite large samples. Then we need, so the reconstruction becomes more difficult if you acquire data, for example, at 20 nanometers and your field of view is maybe 50 microns and your full sample is a few millimeters, then you need to do some extra tricks to not get basically structure noise in the data. But so for example, the zebrafish, they were 6 days larvae and they were whole. We didn't have to cut them.
Great. I'm happy to hear it. And the second question is more technical and is whether for a successful beam time proposal, one would need to show that like benchtop acquisition has already failed? Like whether there's a necessity for synchrotron radiation?
So you do have to convince the panel that you need a synchrotron, but you don't need to show data from lab CT. So at this particular instrument, the resolution is really beyond what can be achieved with the lab source.
Mm-hmm.
So it's more like you need to say, okay, I need this kind of resolution because it's important for my question. So I need this instrument, but you don't need to like show data and to show that it fails.
Okay, thank you very much.
You're welcome.
Okay, I think that let's thank the speaker again. And I think that as we run out of time, I will pass the microphone to Evangelia Christina to continue with the next two speakers. She's co-chairing.
Thank you, Maria. The next speaker is Professor Georgios Panayiotakis from the University of Patras. It's the Department of the School of Medicine, and he's a professor of medical physics.
Good morning again. First of all, I would like to apologize for looking tired, but I have just recovered from COVID. I would like to thank the organizers for giving the opportunity to present my thoughts regarding synchrotron radiation in modern medical imaging. Well, I was saying that I will try to present some thoughts regarding the use of synchrotron radiation in modern medical imaging. Which are the major problems in medical imaging? First of all, that we usually have to image small contrast differences, like breast lesions or microcalcifications. Small sizes, like small vessels. And all these have in mind that we have a very large, a huge number of examinations, and that we have to take into consideration the biological sensitivity of tissues. In other words, we have to balance between the benefit, and that benefit we have in mind the high image quality, and the cost. And that cost we have in mind the radiation does to the tissues. Speaking for a typical X-ray spectrum, like this one, we can have high image quality in this region because we have photoelectric absorption and low scatter. At the same time, we have low radiation dose, in other words, low absorption, in the right part of the spectrum. So we can see that in the middle part of the spectrum, we can have a compromise. Which is the solution? The solution is to work, speaking for a typical X-ray spectrum, in this part of the spectrum, where we are almost monoenergetic beam, but the problem remains regarding the high intensity of the beam. What can we do? We use advanced detectors, new spectra, new geometries. And yes, we utilize all these techniques. Or we can use alternative techniques, not ionizing radiation, like ultrasound, like MRI. But these techniques have a major problem, which is the poor spatial resolution. So, what is next? What is next? Synchrotron radiation. Well, to save time, there is no reason to say what is synchrotron radiation. It has already been explained. I just want to say that we have artificial radiation using synchrotron facilities. And which are the main advantages of this radiation? Is monochromaticity, high density or high brightness, high collimation and high polarization, connected with phase imaging, which are the main limitations. The limited number of facilities, and therefore the difficulty to perform experimental studies, and of course the cost. A few words about our contribution as a research group. In fact, what we wanted to do about 10 years ago is to develop an accurate model to allow in silico evaluation of SR for breast imaging studies. In fact, we were able to do this based on previous work, as we had already developed Monte Carlo tools as group dedicated in breast imaging. Here you can see some of these works, and I'm proud to say that we were the first group in the world to produce Monte Carlo generated mammograms. So, based on this work, we developed a Monte Carlo tool for SR, and we validated this tool, this simulation tool, on Electra SR facilities about 10 years ago. So we had phantoms. I repeat, not human beings, phantoms. Okay. So we validated our simulated work with experimental results. Here we can have a look at some of these experimental studies. Please, I would like to highlight, first of all, here, that the distance is really large. So in fact, we are talking about a parallel beam. What is the advantage of using a parallel beam? That you avoid distortion effects. Please also notice that the half value, the foot was, you can see from here, the full width maximum of the beam, which is about 20 EV only. Here you can see the simulated image and the experimental image. Please notice, I would like to emphasize one point, that here you can see an edge enhancement effect. Why? Due to the phase contrast. You can see also this here at the green edges, at the profile lines. I repeat, due to the phase effect. So we are lucky and proud to have published this validated mathematical tool based on Monte Carlo in the Journal of Medical Physics. In case you don't know this journal, is a high impact journal published by the American Association of Physicists in Medicine. The publication was in 2012. To summarize, synchrotron radiation is gaining ground in the field of X-ray imaging, in the field of diagnostic radiology. For all these advantages that we have already mentioned. Today, we have the ability to have real breast images at Trieste using this system. And in fact, we can use SR North study not only in breast, but in small biological samples, and we have the chance to see such examples in previous lectures. Tomographic imaging and face imaging. We had the opportunity to see such face images, and I repeat, we have the advantage of this edge enhancement effect. Thank you for your attention.
Thank you, Professor Panagiotakis. It's really nice to see that several years ago you have managed to have such nice results from your research. And okay, thank you so much for a nice presentation. I have two questions. The first one is the spectrum that you show is the tungsten anode. So emissions are 60 K, around 60 K. However, the measurements done at the lecture has been done, I think, at 27 K, something like this. So this is well, basically.
If I understood well, our simulated data we used with the same conditions, not with a typical X-ray spectrum, but with a theoretical, let's say, SR spectrum.
Then you mentioned the radiation-induced cancer effects, particularly for the breast cancer. I just wondering, because I heard that the mammography itself, it induced some cancer, and 1 out of 10 is this is the probability. Is that correct?
You know, generally speaking, ionizing radiation is dangerous for the human beings. Always. And unfortunately, there is no threshold. Even a very small amount of radiation can be very dangerous.
Yeah, so you have the statistics updated?
Yes. However, you always have to take into consideration the benefit. What is the benefit? The diagnosis. And the cost. What is the cost? The radiation, let's say, damage. So always, but always, you have three principles. The first radiation protection principle is that all imagists must have a real reason to be taken. Otherwise, you don't take them. Of course, the second radiation protection principle is known as ALARA, as low as achievable, yes. Which means what? Okay, you have to have this examination. You take it under optimum conditions, not only for the patient, but also for the staff and for the general public. And to finish this, there is also a third radiation protection principle, which has to do with the radiation protection of the staff, but it's another story.
I just wanted to know the statistics all the mammography that
I'm not ready to say the very updated data, so, okay. You know, these data are different between countries. And please notice that they are different between ages. In any case, the danger to ignore mammography may be much larger from the danger from mammographic imaging. So, in any case, one of the things that we have to do is to avoid offering fear regarding medical examinations using ionizing radiation. I hope it is clear.
Thank you. Thank you, Professor Maria.
Thank you for the opportunity.
The next talk, which is from Professor Kostomitsopoulos Marketou. The title that she is, from the School of Theology, Religion and Society. The title of the talk is Shedding New Light on Ancient Technologies, Radiation-Based Microbes and Microanalysis of Egyptian food and related mystic workshop of course.
Hi, can you hear me?
Yes, please start sharing your screen.
Okay, can you see the correct screen?
Yes, thank you.
Yes, great. Well, I'm just a postdoc, I'm not a professor, but thank you for the introduction and thank you also for the invitation and taking this initiative. Today I will be talking about some of the possibilities that the ESRF can offer to the fields of archaeology or cultural heritage in general through the prism of my own research experience, just as a user that visited the synchrotron. For a research that was focused on the production of Egyptian blue, which is an artificial or otherwise manufactured inorganic blue pigment that was used in antiquity. Over the last 20 years, an increasing number of heritage scientists, including conservation scientists, paleontologists, as we heard before, archaeometrists, have approached the European Synchrotron Radiation Facility, the ESRF, in order to answer questions related to conservation problems, as we heard, art history or archaeology. There are several reasons that may have contributed to this increasing need to approach the ESRF for such research questions. And perhaps the most crucial one, compared to the laboratory-based analytical techniques, is the possibility that the synchrotron offers to study minute samples, very small samples, in a high resolution of analysis, something that is particularly important for samples of cultural heritage, which are unique, irreplaceable, and for which often curators would allow only non-destructive or non-invasive analysis. The materials of interest for the researchers in the broad spectrum of cultural heritage can be divided into two categories: the manufactured or artificial materials, and the natural or non-artificial, non-manufactured materials. For archaeology, which is, in my opinion, maybe one of the most diverse scientific fields, materials from both categories are important and can be used as proxies to approach complex questions regarding the organization of ancient societies and their relationship with the environment, the relationship of people with the environment. A range of materials have turned towards the bright light of the synchrotron over the last years, from blue-colored bone materials to complex ceramic firing processes of the so-called Berlin painter from ancient Athens, and from the monk's scream to ancient Egyptian inks applied on papyrus, just to list a few of the recent publications. So several applications have contributed with new knowledge to complex archaeological and conservation science questions through the use of a range of analytical techniques. Today's talk, of course, does not aim to cover the richness of the research carried out at the ESRF, and this has already been done excellently from the colleagues talking before me, and that will continue talking after me. And since I am not an expert myself in the use of the synchrotron, my talk will focus on my experience with working there for a specific experiment at the ID21 beamline, focusing on the work on the production on Egyptian blue. My presentation aims to guide you through the methodological process that we followed based on specific research questions, and aims also to highlight the significance of techniques such as those offered at the ESRF for and the cultural or historical interpretations that such hard data can give insight to. The experiment at the ESRF was part of my PhD research project that I carried out at the Department of Archaeology, Conservation and History of the University of Oslo in Norway, and it aimed to approach the material evidence of pigment production, the production of colors for those for the wider audience, that were found at a late Hellenistic workshop on the island of Kos. That is an Aegean island in the Dodecanese complex. The project focused on the materiality of the finds and framed the pigments as artifacts, i.e., the intentional outcome of a series of technological processes. So what is Egyptian blue? Egyptian blue is considered to be the first artificially produced pigment, manufactured pigment. It can't be found in nature. And it was first produced in the 3rd millennium BC, and it was used continuously for about 4 millennia. This is a multi-component material that is produced by firing a mixture that essentially contains a copper, a silicon, and a calcium-containing compound, together with an alkali flux in high temperatures from 850 to 1050 degrees Celsius. Its primary compound, the one that gives the material its blue color, is a copper-calcium tetrasilicate crystalline phase that is equivalent to the naturally occurring mineral known as cuprorivide. The production site of Kos was excavated by the Ephorate of Antiquities of the Dodecanese, the Greek Archaeological Service, and the archaeologist Haris Kansia in the city of Kos. In the 80s, the excavation period lasted for about 5 years. The workshop is located in the Stoa complex that runs along the eastern border of the Agora of Kos, an area that was known to host commercial and manufacturing activities in that period. There, more than 100 Egyptian blue pellets were found, together with earth pigments, and also the remains of metallurgical processes. As you can see in this photo, the Egyptian blue pellets that were found at the workshop vary in terms of color from saturated blue to green, gray, and even purple. The study of the Egyptian blue pellets began with a macroscopic observation of the finds at the storerooms of the Ephoria, and they're divided into 3 groups based on the surface color of the finds. So the successfully produced pellets had a saturated blue color, while the unsuccessful pellets had anything from gray to brown, and the partially successful pellets were those that had blue observable on their surface, but they also had other colors, mainly green. We then proceeded with a preliminary elemental analysis of a selection of pellets with portable XRF, and this allowed us to cluster them in groups based on their elemental composition, aiming to see if there is any relationship between the elemental composition of the finds and their final appearance. And finally, we proceeded to sampling of selected pellets. We took 19 samples and we examined from different pellets that had different colors macroscopically, and we examined them with scanning electron microscopy coupled with energy dispersive X-ray spectroscopy, anyway, SEMI-DS, and micro-Raman spectroscopy. The combination of this analysis indicated that there is a high level of expertise in the production process and a deep knowledge of the material transformations that were involved in the production. In other words, the chemical processes that were involved in this inorganic synthesis. And that the craftspeople that were working on COS not only knew how to produce the initial material, but they could also further process the blue pellets, like this one, to produce different hues and tonalities of blue. So we have these people that we know now that they actually had high technical knowledge and chemical knowledge, let's say, that were working in the 1st century BCE. And they knew how to produce Egyptian blue, but at the same time, we have about 1/4 of the pellets that have this outcome, which we call unsuccessful production. This observation puzzled already Haris Kantia, the excavator, and Kelly Kuzeli, a chemist that is working for the Ministry of Culture, already in the late 80s. And they analyzed 2 pellets, one successfully produced and one unsuccessfully produced, to try to figure out what the problem was in the production. What they found is that the unsuccessful production has a high level of iron content in the final product. Okay, so here comes the new research that was carried out on the exact same pellet, which we took since it was already sectioned from the previous analysis. And we took 4 samples. As you can see, the pellet varies from brown to green and blue. So we took 4 different samples from this sectioned pellet. We embedded them in resin and created cross sections. We approached the ESRF. Our team, consisting of myself, Francesco Caruso, Francesco Giannizzi, Soren Handberg, visited the synchrotron, where we found Wout de Nolf and Marine Cot at the ID21 beamline. And we worked there for 3 intensive days, carrying out micro-XRF and micro-XANES analysis. From each one of the 4 samples, we retrieved the elemental micro-XRF map for copper, iron, and calcium. And based on these maps, we selected the spots. As you can see, these numbers are each for 1 spectrum for the XANES spectrum. We also analyzed the standard copper compounds and commercially available Egyptian blue samples. And then we carried out principal component analysis on the normalized XANES spectra from the archaeological samples and also from the reference materials. This analysis aimed to highlight the possible clusters or outliers in the dataset and to determine if we had different phases of copper compounds that could give us insight into the production process, into the details of the production process. As you can see in this graph here, all the Egyptian blue samples were clustered in this rather large cluster. We argue that this is because of the variation of the crystal orientation of the Egyptian blue crystals with respect to the microbeam. The few outliers seem to correspond to the crystalline phase of dioptase. And in general, we didn't have any, we only had bivalent copper phases, which was important, as you will see in our conclusions. So, by combining synchrotron-based micro-XRF, micro-XANES, and the laboratory-based analysis that we carried out, including optical microscopy, ICM-EDS, and micro-Raman spectroscopy, we were able to determine most of the phases present in the complex matter of Egyptian blue. And the fact that even in the unsuccessful production, we mainly had cuprarivite crystalline phases was interpreted as an indication that throughout the firing, we did have sufficiently high firing temperatures. And this means basically that the unsuccessful production was not an outcome of an insufficient firing. Moreover, the fact that we have predominantly bivalent copper compounds in our samples point to an oxidizing atmosphere while firing, and therefore the hypothesis that reducing atmosphere would disturb the production process was not validated. Rather, the results indicate that the cause of the failure in the production is probably limited to the starting materials, which most likely included iron compounds as impurities, and to a lesser extent, perhaps to poor mixing and grinding of the materials. This means that basically, even though the people had the high technical know-how, the materials, the raw materials that they were using for the production were not standardized, as we can imagine in the first century. And this would lead to a randomness, let's say, in the production regarding the successful or the unsuccessful outcome. So I'm coming here to an end. And I just wanted to mention that this work that started from the excavation about 40 years ago and the formulation of the first research questions regarding the production technology of Egyptian blue was always in close collaboration between archaeologists and natural scientists, starting with Kantia and Kuzeli. And we have indeed come a long way, throughout which several research questions have appeared and still appear. These are some of the publications that came out from my PhD work on the workshop. And all of this couldn't materialize without the work of all the researchers that I have mentioned in this slide. And I hope I'm not forgetting anyone, who were working in different institutions in different countries. And this work, the work that we did at the ID21 beamline of the ESRF, was definitely a very important step in the process of exploring the materiality of Egyptian blue from the COAN workshop that shed, I hope, new light to the highly complex technology of ancient pigment production, and answered unanswered research questions related to the copper compounds that could not be answered in conventional lab techniques. Thank you for your attention.